Plants go through a process called photosynthesis to make energy. They use carbon dioxide, water, and light to produce glucose and oxygen. Photosynthesis begins when light pigments in chloroplasts absorb energy from the sunlight within a narrow range of wavelengths. The pigments in the wavelengths include chlorophyll A, B, and carotenoids. When the light is absorbed by these pigments, the energy from the light is then integrated into electrons within the atoms, and when they absorb that energy, they get excited and move up to a higher energy level. This energy harnessed by the plants is then used to generate ATP and NADPH.
So our experiment will be testing how putting a red film over a beaker of leaf discs will affect the rate of photosynthesis. We'll be conducting two trials, one without the red film on the beaker and one with the red film on the beaker. Our independent variable is the red film, and our dependent variable is how this affects the rate of photosynthesis. We predict that the rate of photosynthesis will be slower with the red film on the beaker because the film is reflecting red wavelengths, which are used for photosynthesis.
The data in the lab consists of the control and the experimental, which uses red foil to change the wavelength of light. The data showed that the light directly from the lamp would let plants photosynthesize faster than the one that only let red light in. Specifically, it took a total of 11 minutes and 26 seconds for the 5 discs in the control to photosynthesize, but took 14 minutes and 30 seconds for the discs in the experimental to photosynthesize. This led us to the conclusion that the control had more energy for the plants to absorb since it photosynthesized faster. The graph of the control had a slope of about 0.44 discs per minute, and the graph of the experimental had a slope of about 0.35 discs per minute. This showed that there is about a 20% difference between the two photosynthetic rates, and with this information we can conclude that a major source of energy for plants comes from red light, since the red light had a difference of only 20%. We can confirm this with many other articles we have read that have stated that red light is one of the most important lights for plants due to its abundance. The experiment's data simply proves that red light is a key factor in photosynthesis with the use of discs.
So the hypothesis proposed that if all colors of light except for red are filtered out from the light source for the photosynthesis of the leaves, the rate of photosynthesis will slow down because there would be less wavelengths of light for chlorophyll to absorb, thus slowing the rate of absorption and photosynthesis. The data revealed that putting a translucent red screen over a light source increased the estimated time for 50% of the leaves to float, ET50. There was some support for the hypothesis because a longer ET50 indicates that the rate of photosynthesis is slower. The data showed that the control trial had an ET50 of 11 minutes and 26 seconds, while the photosynthesis under the red filter had an ET50 of 14 minutes and 30 seconds. Removing most of the other wavelengths except for red resulted in an increase of 3 minutes and 4 seconds in ET50. Moreover, the average rate of change of the number of leaves in the control trial was about 0.3417, while the average rate of change for the number of leaves that floated in the red light was about 0.3167, further demonstrating that rate of photosynthesis slowed down in the trial with filtered light. The results of the experiment show that if wavelengths of light are removed from the source for photosynthesis, the rate slows down. The results make sense because many of the different types of absorption pigments, such as different chlorophylls, are not activated to facilitate the light-dependent reactions of photosynthesis. Furthermore, when certain wavelengths of light are removed, there is less overall energy being taken up by the plant. Therefore, less oxygen would be produced by the plant to be stored in the mesophyll tissue, and the leaves would not be able to float as quickly. The following confounding factors, transparency of the filter sheet, amount of initial oxygen in the mesophyll layer, and the effectiveness of the light source could have affected the outcome by influencing the rate of photosynthesis. If the filter sheet did not adequately reflect other wavelengths of light, it would not be clear if the color of light was changed between the trials. Similarly, if the light source did not give off equal levels of each wavelength, it may have affected the results because the filter would not be as effective. Moreover, if not all oxygen was depleted from the leaves prior to being placed in the sodium bicarbonate solution, then the leaves may have floated more quickly. Additional research questions such as how colors of light other than red affect the rate of photosynthesis may clarify how wavelengths of light affect photosynthesis.